Hello students, in this lecture we are going to discuss about the structure of the chromosomes, which is an integral part of the basic studies towards genetics in our class 10th syllabus. So what are chromosomes? The chromosomes are in fact described as colored bodies. So chromosomes were for the first time observed by Strasburger and Fleming in animal cells. The term chromosome was given by Waldeyer. Basically, the term has been taken from a Greek word known as chromosoma, means colored bodies, because of their marked ability to take up basic dyes, for example, acetylcarmine and fullerene. So, technically, if we want to describe the chromosomes, so we can call them on the basis of a staining pattern, we can call them as the darkly stained rod-shaped bodies visible under light microscope in a cell during the metaphase stage of mitosis is known as chromosome, or these structures are referred to as chromosomes. Another definition says that chromosomes can be considered as a stainable thread-like nuclear component having spatial organization, individuality and function. Another definition is the chromosomes are simply colored thread-like structures observed in an actively dividing cell. So you can write either of the definition, all are considered to be correct at the level of class 10th. But can we expect to observe the chromosome in a cell? Definitely not. The chromosomes are ordinarily not observed in the interphase nucleus or when the cell is in the steady state or when the cell is resting. That means it is not actively dividing. Then you cannot expect to find a chromosome. Now, in order to understand the structure of chromosome, we will discuss about the structure of DNA and we will discuss about the organization of DNA around specific protein molecules. So first of all, let us try to understand the structure of DNA with special reference to chromatin. So DNA is major, we all know that DNA is the major genetic constituent of the chromosome. Without DNA, there is no question of existence of chromosome even. So histone protein along with the DNA, the double helical DNA, is known as a nucleosome. A nucleosome consists of 8 molecules of proteins along which the DNA strand is wrapped. Okay, so if we observe DNA along with histone proteins is commonly known as chromatin. If we observe the chromatin carefully, it appears beaded. Why it appears beaded? Because of the presence of the pack of histone proteins along with the DNA. It is technically known as nucleosome. So this nucleosome or the beaded thread of the chromatin together forms the complex structure of chromatin thread. This chromatin thread undergoes a series of coiling and supercoiling. This results in the formation of the detailed or the exhaustive structure of chromosome. Imagine if the entire DNA is opened from end to end, then how much length it will take? It will take a length of approximately 2.4 meters. Think how complex is the coiling and supercoiling and how efficient is the packing, that the entire 2.4 meter long DNA is contained in a structure that is visible only under the microscope. Watson and Crick, they said that the DNA is very much like the spiral staircase. Each DNA molecule consists of repeated sequence of nitrogenous bases associated with pentose sugar and phosphate group, and these are known as nucleotides. The nitrogenous bases are classified into 2 major categories: purines and pyrimidines. The 2 purines are adenine and guanine, and the pyrimidines are thymine and cytosine. There is one more nucleic acid known as the ribose nucleic acid. In that case, there is an additional nitrogenous base, and this nitrogenous base is known as uracil. Along with that, there are phosphate and sugar backbones which provide additional stability to the DNA molecule. If we talk about the gross structure of the double strand, so they are considered to be anti-parallel to each other. Chain is known as the 5-3 chain and another one is known as the 3-5 chain. The 5 3 chain is known as 5 prime end, and the 3-5 chain is known as the 3 prime end chain. So it consists of the turns. Each turn consists of 10 nucleotides, and it is separated from the another turn by a distance of 3.4 nanometer. Okay, so adenine pairs with thymine, guanine always pairs with cytosine. It is known as complementary base pairing. We will discuss about the further details, and more elaboration on genetics will be followed in the next lecture related to the division of the cell. So if you have any questions, you can put it up. Thank you for being patient and listening carefully.